So it's my great pleasure and honor to be here today. So, and also I want to thank you, the organizer of this AVI Open Science Forum, to give this opportunity to present our research. Today, I will briefly introduce our UNAPP Center, just as Alpha introduced, and also I will trying to later I'm trying to dive into a specific topic like my team has been leading on the dengue virus antiviral development by focusing on targeted non-structured protein 4B for Flavivirus therapy. Okay, let's move on the slides. Okay. So to begin with, I want to share a little bit with you about my research background. So I started my PhD working on dengue viruses and antiviral drug development. And I was very fortunate to have an opportunity to receive training in the Novartis Institute for Tropical Disease, NITD, in Singapore, which was then a global leader in antiviral drug discovery for dengue and other neglected disease. And then in 2016, I joined Dr. Xuping Xie's team at UTMB for my second postdoc, and then became a faculty member at UTMB. So while the Zika outbreak in 2015 and 2017, and then later COVID-19 pandemic briefly shifted our focus, actually, I have been since returned to dengue viruses research as part of the UNAPP initiative in 2022. So my lab currently focuses on three main pillars of research. One, we want to develop, we can development of reverse genetic system for positive sense RNA viruses, including infection clone, reporter viruses, and high throughput assays. Those are critical tools for research. And we were among the first lab to develop a reverse genetic system for Zika and SARS-CoV-2. And the second, we are very interested to understand some biology about such as the molecular mechanism of virus replication, assembling, evolution, and host viral interaction that influence pathogenesis. And the third part of our research is focused on develop anti-counter measurements such as vaccine, monoclonal antibody, and antiviral drugs, often through academic and industry partnerships. So for those who may not know, UTMB has a long legacy in infectious disease research. Since its founding in 1891, it has been at the forefront for measure public health responses. So the establishment of Galveston National Laboratory in 2008 actually further expanded its capacity, so providing all kind of state-of-art biosafety laboratory 2 to biosafety laboratory 4 for and animal facilities for research and education. So the GNL has played a critical role in tackling outbreaks of Ebola, Zika, and SARS-CoV-2, Nipah, and the most recent H5N1 and the measles as well. So to this group, I think I don't need to mention too much about how many emerging or re-emerging infectious diseases that have been seen in the past, and I'm sure we are going to face more in the near future. So, and the best defense, is preparation, right? Through knowledge, infrastructure, and the medical countermeasures. And as like United States founder father Benjamin Franklin say, so by failing to prepare, you are failing to fail. So I think we should get ourselves prepared. With this in mind, UTMB joined the force with United Service, United States University, Emory University, and Novartis to form UNAPP in 2022. So we have a unique academic industry partnership. So our mission is to develop antiviral drugs that targeting coronaviruses, Nipah and the Flaviviruses. As you know, many of them are significant human pathogens with unmet medical needs. So we are focused on small molecules in our center. So we have built a cohesive team consisting of 4 functional cores supporting overall 7 projects within this industrial-grade antiviral drug development pipeline. So our overarching goal is to deliver late preclinical stage antiviral candidates that can be rapidly advanced in the future in case of outbreak or pandemic. So I'm leading the virology core and also the focus on the mechanistic study of dengue viruses and 4B inhibitors. So in this rest of the talk, I will share our recent findings regarding the antiviral let me pull my point. Antiviral mechanism of dengue NS4B. In our center, we have a large program focused on the flaviviruses using multiple targets such as NS5, NS3 protease, and NS4B. So to those who may not be familiar with dengue, dengue caused by dengue viruses is the most important mosquito-borne viruses affecting humans and has been endemic in over 100 countries. And nearly half of the world is at risk of dengue each year. So dengue causes 400 million infections, 100 million illnesses, and 40,000 deaths. In the past 2 years, we have seen a huge surge in the Americas, at 2023, 2024. And currently, vaccine and vector control has some limitations. We don't have any antivirals or therapeutic antibody can be used to treat dengue patients. So the dengue disease symptoms range from mild fever or flu-like symptoms and also severe disease such as hemorrhagic fever and the dengue shock. So I have never forget things, seeing children suffering from dengue, severe dengue rashes showing here. So during my visit to a hospital in Philippines in 2009, so during my PhD, so unfortunately, not much has changed since. So severe dengue often arises during secondary infection by heterologous serotype due to antibody-dependent antibody enhancement, ADE for short, which also complicates the vaccine development. This table just shows you a countermeasure landscape about for dengue. So unfortunately, beyond the limited vaccine, Sinovac vaccine being approved for patient for use. So we don't have any other molecules or a small molecule antibody or small molecule vaccine or antibody available. So two molecular antibody and also two NS4B inhibitor highlighted in the green here are in clinical trials, but they are not yet approved. So a very brief about the basics of dengue viruses. So compared to coronaviruses, so dengue virus appears relatively simple. So it contains a single strand positive sense RNA genome that encodes 3 structure proteins CPIME and also 7 non-structure protein NS1 to NS5. So the non-structure are key to virus replication and assembling and immune evasion, while the NS3 and NS5 are the major enzymatic target that have been used for antiviral research. But despite efforts of decades of efforts, it has been proven there's no low-hanging fruit for these antiviral targets. So NS4B, which is a new emerging kind of target, have been gained attention as I mentioned earlier. So our research center has focused on explore NS3 and NS5 and NS4B for dengue antiviral in parallel. So very encouraging, actually, significant progress has been made in enhancing NS4B inhibitor potency across all 4 serotypes during the past 10 years. So for example, the first dengue inhibitor, NITD 618, I worked in Novartis when doing a PhD. So it's only had 1 micromolar efficacy. Now we have seen NITD 68 and GAG compound 2 with nanomolar or even picomolar potency, and now in phase 2 clinical trial. So you will hear more on both of these inhibitors through my presentation later. So they have very different antiviral mechanism I'm going to show you in a couple of minutes. So a very brief introduction of the NITD 68. So before I left the Novartis, our Novartis colleagues discovered a compound showing here, the compound 1 from the robotics chemical library using high-throughput cell-based screening. So after very simple quick optimization, NITD6A was made. So NITD6A has drug-like property and the nanomolar efficacy against all serotypes in vitro, and the drug also have effective in animal models. However, there are some limitations such as moderate oral bioavailabilities, high blood partitionally, and also high plasma protein binding. So furthermore, we don't know much about this mode of action. So within this scope of our center, we have a specific aim by trying to address this liability as well as gain antiviral insights about this antiviral mechanism of this compound. Today, I will mainly focus on discuss the first part of understanding how NITD6A works at the molecular level. So this work was led by my postdoc Yan Wang, a talent in my lab through resistance selection, showing here after 15 passage, we identify some critical mutations in the NS4B. So they are sitting on the ER membrane or the lumen side. We engineer this individual mutation or combined mutation using infectious clone of dengue 2. So we confirm they are resistant to this compound, as we can see the increased EC50 values. So this confirmed the NS4B is likely target of NITD6AA. So this is showing you three key mutations. So to further determine whether NITD6A directly bind to this protein, so we measure the binding affinity using purified NS4B protein, and we using a technology called isothermal titration calorimetry, like ITC for short. As you can see here from this table, NITD6A binds tightly nanomolar KD to all serotype NS4B, and then locally the mutations we identify from the resistance selection significantly reduce the binding affinity. And this is consistent with our previous MR data, specifically showing here the wild-type NS4B after NITD6 bind, showing chemical shift with a mutation, so we don't identify any shift upon in the presence of NITD6A. So it's very well known that NS4B interacts with multiple viral and host proteins. So using pull-down experiment, we want to identify whether what's the impact of the compound binding to those interactions. So we found that, long story for short, we found that NITD6A specifically disrupt NS4B NS3 interaction in a dose-dependent manner. As you can see, at the 5 to 100 nanomolar NITD6A drastically inhibit the amount of NS3 pulled down by the NS4B, shown here. So importantly, we also use mass spec showing no significant off-target effect on host protein interactions. The interactor pulled down by NS4B in DMSO treatment or in the compound treatment were largely similar. So these results demonstrate that NITD6A primarily inhibits NS4B NS3 interaction. So a recently PNS study from Dr. John Caritis from Stanford University further confirmed that the NITD6A minimally perturb the post-transcription of, as you can see, very few genes were impacted by the treatment of NITD688. This is similar to NS5 inhibitor, NITD008 nucleoside analog inhibitor without target other lone host protein, also showing minimal impact on the gene expression level. However, the other NS4B inhibitor, GNG compound, affect over 500 host genes, suggest like broad off-target effects beyond the NS4B by GNG compound. So we want to ask how does that molecule work in the molecular level, right? So we next use two technology biophysical assay to examine how NITD6AA disrupt this interaction. So on the top panel, using ITC experiment. So we are increasing, you can see, focus on this panel. So increasing concentration of NITD6A, we're showing a decrease the NS3 binding to the NS4B. So indicate the NITD6A prevents this NS4B, NS4 formation. I want to point out here, the NS4B was pre-treated with compound before they mix with NS3. So in the second experiment, we kind of using a biolayer interferometry assay called BLI for short. So in this experiment, we preform the complex of NS4B, NS3 on the biosensor. Then we treated this complex with different concentration of NITD68. As you show from this dissociation curve, NITD6 promoted this complex dissociation in a dose-dependent manner, demonstrating this NITD6A can also disrupt these performed NS4B, NS3 complexes. To further evaluate whether this mechanism also applies in cells, so we performed pull-down experiment in the 293T cells using co-expression of NS4B and NS2B, NS3. So we added a compound either at 4 hours, represent the early treatment when during the formation of NS4B and NS3 complex, or 24 hours kind of delayed treatment, represent that NS4B and NS3 complex and be formed. So in both conditions, we're showing here in this graph, the NITD6 showing in blue bar, so significantly reduced NS3 pulled down by NS3 in both 4 hours, 24 hours. In contrast, JJ compound only showing effect at the early treatment. So these results indicate that the NITD6 is different from JJ compound 1802, so can disrupt preformed NS4B and 3 complex in cells as well. So we next want to ask whether this is happening also in infected cells, right? So to do this, we designed an antibody probing experiment using 2 in-house generated antibody called 4447 or 1037. So they target different epitopes. So 447 target the epitope within the cytosolic loop overlapping with NS3 binding site. And the monoclonal antibody 10237 targets the N-terminal region epitope does not affect by NS3 binding. So during the infection, so NS3 binding going to mask the epitope of 4447. So we hypothesize that the NITD6A treatment going to disrupt this complex and then will release this epitope with enabling detection by the 4477. Right, to do this, we're using a delayed treatment experiment. So HU7 cell here were used, in fact, with dengue viruses at high infection MOI, and inhibitor was added 20 hours post-infection. So next, after 16-hour treatment, we fix the cells, and then we're standing by different antibody, NS4B and NS3. So we're using NS3 as a control for internal normalization. As you expected, so NITD6A significantly reduced the NS4B level, showing here in the infected and the treated cells, and also in compare with DMSO treatment of NITD6A. Surprisingly, showing much brighter NS4B standing with by standing 4447 antibody suggests this epitope unmasking due to the NITD6A treatment. In contrast, so we use a 10337, we confirm that NITD6A can potently inhibit both NS4B and NS protein expression. So what's more, we're also testing other inhibitors, as you can see here. So only this is phenomena also only specific to NITD6A8 have not been detected in other NS4B inhibitor JJ compound or NS5 inhibitor NITD008. So then we want to ask what this does this have any biological significance? So we performed a more comprehensive time of addition study using a dengue reporter viruses. So early treatment will kind of likely targets complex formation, while late treatment going to target, the preformed complex. So consistent with the mechanism of NITD68. So this compound showing minimal loss of potency, as you can see, from 1 hour to 24 hours, similar to NITD008 compound. In contrast, GAG compound, you can see particularly 16 to 20 hours post-treatment, GAG compound efficacy was markedly reduced at the late time points. So this demonstrates that unique mechanism of NITD68 has potentially, was less impacted by the delayed treatment compared with the GAG compound. So this come to our working model. So it's very long, this NS4B and 3 interaction. So it's critical for virus and replication. This has been reported by our group previously and also by Dr. Ralph Butenshanker's group several years ago. So the NITT compound, as I showing before, is combining to NS4B, and we think because they're binding on the other side of this membrane. So we believe this can induce a conformation change of the cytosolic loop and disrupt this NS4B and 3 complex and in the infected cells and consequently halt the virus replication. To further validate this mechanism, we engineered, this critical mutation use another virus, which is a Zika virus as a model. So Zika virus showing here doesn't have no sense, is not sensitive to NITD68 treatment. It's very interesting once we're including this NITD68 binding residue in the T197 or A2224. So they confirm this sensitivity to compound NITD68. Using same assay, ITC assay, we confirm this compound can bind to the NTD6 can bind to the Zika NS4B mutant with these 2 mutations, but not the wild type, as shown by the KD value. And more importantly, so using the same ITC method, we determine, so in the presence of the NITD68, NS4B mutant interaction with NS3 has been reduced. So using this system, basically we confirm, not only confirm the mechanism we have been showing previously, and also support the strategy of targeting NS4B, NS3 interaction for Zika antiviral and development. So as a conclusion, so I want the UNAPP, our center is a unique public-private partnership that can accelerate drug development for pandemic preparedness, and we continue to support is essential to strengthen our resilience against the future outbreaks and the pandemics. So I hope I convince you, we have given you critical insights about antiviral mechanism of NS4B inhibitor NITD68 targeting the NS4B NS3 interaction. So you can find more further details in our recent publication on the PNAS journal. In the near future, we want to continue this work trying to obtain high resolution structure of flaviviruses NS4B and complex of NS3. And also we are trying to explore the potential combination therapies such as this inhibitor with NS5 nucleoside analogs or NS3 protease inhibitor. I think Darlene will be mentioned later. So, and also lastly, we want to apply this strategy to target, basically target NS4B and NS3 interaction for developing antivirals against other flaviviruses such as Zika virus and the West Nile viruses. So finally, I want to thank you, all my team members at UTMB, and also my collaborator from different department of UTMB, and also my collaborator with Emory University, Vinny, particularly Vinny and Manachari, and also the USU, as well as my long-term collaborator and also our UNAPP team members from Novartis, Thierry Daglan and Lydin and Joris and Gufeng and Colin Skipper and Stephanie and also other members within team provide their invaluable contribution to this project. And also, we have been very grateful to acknowledge NIH and other foundations for financial support. And thank you all for your listening, and I'm happy to take any questions.